Good afternoon, everyone. On behalf of the Faculty Interactions Committee, I'd like to welcome you to the fourth presidential lecture, this year hosted by Dr. Ozawa and Dean Tomaselli. The presidential lecture was initiated by the Faculty Interactions Committee to highlight the interactions between the Einstein-Montefiore faculty that reflect outstanding basic and clinical research that often highlights the collaborations between Einstein and Montefiore. The first lecture was given in 2017 by Drs. John Condelis and Joe Sperano on their long-term collaboration on identifying roles of the microenvironment in breast cancer progression. In 2018, Drs. Betsy Herold and Bill Jacobs gave lectures on their groundbreaking work on developing new vaccines against the herpes virus. In 2019, we heard about the exciting basic science to bedside work of Drs. Joan Berman and Chinazo Cunningham. Today, after a pandemic break of 2 years, we will hear about the exciting collaboration between Drs. Uli Steidl and Amit Verma on stem cells and cancer of the bone marrow. Congratulations to Uli and Amit on their selection by the Faculty Interactions Committee from an outstanding field of nominees. And now, without further ado, President Ozawa has some introductory remarks to make, and then Dean Tomaselli will introduce Dr. Ulrich Steidl.
Thank you. Thank you very much, Dr. Sharon. And first of all, thank you for more than 20 years of innovative research at Einstein. Thank you. And thank you to our Dean Tomaselli, for supporting this lecture series and shining a light on the seamless bond and the translational nature of research. I'm also grateful to all the members of the Faculty Interactions Committee for making this presidential lecture series possible. You know, Yo-Yo Ma said that a really good piece of music is more than just itself. He said, it's like a prism. From each facet, you see a single totality. The same, I believe, can be said for the 15-year partnership and collaborative spirit between Dr. Steidl and Verma. They arrived here just about a year apart, one from Heidelberg, the other from New Delhi. However, the desire to bring laboratory findings quickly to clinical application precipitated a partnership and a relationship that ever since has added immensely to our understanding of a group of deadly and yet intertwined disorders. You know, science is often depicted as a lonesome endeavor. The single-minded researcher charting new territory into the unknown, one experiment at a time, and often alone. But if you've been at Einstein for even 5 minutes, you know that science at Einstein is particularly unique because it is truly a cooperative and collegial and collaborative endeavor. And because these two brilliant collaborators have played their instruments with such harmony and for so many years, Dr. Steidl and Dr. Verma have moved us closer to the day when we might see the single totality of this group of awful afflictions, and closer to the day when we might end them. You know, Amit and Yuli, I've said this to each of you privately in the past, but I could not be more proud of what you're doing and of your work, and proud of you for being role models for our quest for the full iteration of that Montefiore and Einstein prism, and our quest to get to a day when one would look through any of the facets and see that single totality. I'm proud of you.
So it's great to be back together for a presidential lecture. I want to thank everybody for coming. I want to thank Dr. Oswa for his comments, Dr. Sharon for really hosting today. And I also want to thank somebody who's not here, Dr. Pamela Stanley. She's been instrumental in this lecture series, and the concept of the presidential lecture series, as well as the faculty group that worked with her to make this happen. As has been mentioned, I think our speakers today represent the power of collaboration between Einstein and Montefiore. Working together really has a multiplier effect on productivity, on impact, and the satisfaction with which we do our work, along with the ability to ask incredibly important and clinically relevant questions. So Uli and Amit, congratulations, and thank you for all you've done for Einstein and for Montefiore. It's now, I'm not going to belabor you with a lot of comments. It's my pleasure and my honor to introduce the speakers today. So let me start with Ulrich Steidl. Dr. Steidl is professor of cell biology and medicine, the deputy director of the Montefiore Einstein Cancer Center, and interim director of the Ruth L. and David S. Gottesman Institute for Stem Cell Biology and Regenerative Medicine. Uli earned his MD and PhD degrees at the University of Heidelberg and the German Cancer Research Center. He's fully trained in hematology, oncology, and clinical immunology, and did so at the Heinrich Heine University Medical Center in Dusseldorf before making the leap to a research career. He was a research fellow at the Harvard Stem Cell Institute, Beth Israel Medical Center, and subsequently was promoted to instructor of medicine at Harvard Medical School. We were really fortunate to recruit him in 2008 to come to Einstein, and he quickly rose through the ranks to become full professor, and along the way had a number of leadership positions in the Cancer Center and the Gottesman Stem Cell Institute. His research has focused on the earliest origins of myeloid malignancies, and he's made fundamental discoveries, particularly in the area of precancerous stem cells and transcriptional dysregulation. In close collaboration with Dr. Verma's laboratory, Dr. Steidl's group has identified novel targets and drugs directed at precancerous stem cells in myelodysplastic syndromes and acute myeloid leukemia, which have made it and been translated into the clinic with the ultimate goal of prevention of these deadly diseases. His work has been recognized by numerous honors, including being elected to the ASCI, an Outstanding Investigator Award from the National Cancer Institute, and he is currently the president-elect of the International Society of Experimental Hematology and will assume the presidency later this year. So I welcome him to the podium to give his talk, Understanding Molecular and Cellular Pathogenesis of Myeloid Malignancies at the Stem Cell Level. Uli, thank you.
Great. Thank you so much. first of all, Dr. Ozua and Dr. Tomaselli for this extremely kind and generous introduction. And I also want to first really thank the selection committee for selecting us today. It's a great, an enormous honor for us and for me to speak to you today. And I want to give you in these 20 or a little over 20 minutes that we have a little bit of a run through of some of the work that we have conducted in the last 15 years or so. So, you know, this is basically a model of the pathogenesis of leukemia, like we would have drawn it about 2 years ago. And I would like to emphasize that really we've moved considerably in the last 15 years from understanding that there's not only a tumor bulk, and that's a principle that has also spread, of course, to many other, including solid tumors in the meantime, but that there are so-called leukemia, in the case of leukemia initiating cells that are really at the root of the problem. But that also these leukemia initiating cells, they don't occur out of the blue and overnight, but they are the process of a multi-step transformation process, and they go through various stages that we and others early coined pre-leukemic stem cells. And you know, all these cell types are very important and of clinical importance for a variety of reasons that we'll discuss today. They are an important source of tumor heterogeneity, of leukemic progression and relapse, but also interestingly, some of them are actually compatible with healthy aging, you know, in a phenomenon of clonal hematopoiesis. And it's more blurry than ever, you can say, you know, what is really a normal stem cell? What is a partially mutated stem cell? What is a stem cell that's on its way to produce a cancer? And, you know, that's the theme complex that we've worked on in the last 15 years. And we've made some early contributions here and really went back and forth, as you'll see throughout my talk, between laboratory models, mouse models, studies in patients with human individuals with MDS and AML, etc. And so I want to just go back for about 11 or 12 years when Britta Will, at that time in the lab and closely working with Dr. Verma and myself, you know, way before she became the famous independent PI she is today, showed for the first time clearly and cleanly that stem cells in patients with MDS carry both genetic and epigenetic aberrations, and also that these stem cells actually closely correlate and predict the clinical course of the disease. And Dr. Verma will talk about this more. And then Britta and we went to the mouse and developed the first mouse model of pre-MDS stem cells. This was in collaboration together with Winfried Edelman and of course, again, Amit Verma, and developed the first mouse model that had pre-MDS stem cells and then went through a stage similar to the human disease, MDS, and then different types of mature, fully transformed acute myeloid leukemia. And then a few years later, again, we took it to the next level and switched back to the human system. And a very talented graduate student at that time, Jahao Chen, performed targeted deep sequencing of single stem and bulk cells from individuals who were first diagnosed with MDS, but then later progressed to secondary AML. And in this process, he found, and we found very quickly, that the old linear model from a stem cell via clonal hematopoiesis to MDS and AML, you know, did not match the experimental data that we found, but rather than that, what we saw was a pattern of a nonlinear parallel polyclonal evolution, in that the bulk of the disease, of the MDS disease, was derived from different stem cell subclones and subsets than what would later progress to an AML. And, you know, so this obviously has implications for our understanding for bulk cell focused precision oncology approaches. And so this is a more current model, and that's from a recent review article that we wrote in collaboration with John Greeley here. And what you can see here, that we now think the disease of leukemia and myeloid malignancy is really a polyclonal process from very early on, with a lot of stem cell subclones pre-existing. Therapy is pushing back at some of them, then others that are already there, they don't need to be formed. They just simply emerge through the selective pressure. You apply again therapy, yet another subclone comes out. Some of these small subclones at the stem cell level are below the clinical detection limit, so you can only see them when you specifically look for them in the stem cells, but nonetheless, you know, they are there and cause a lot of clinical problems in the process. And so this is now the accepted model, so really not a linear process in the progression, but that these pre-leukemic stem cells are a very heterogeneous bunch to begin with, and that different clones get selected for at different disease stages. You know, again, we have shown this for MDS progression. Other groups, including John Dick's group, have shown this for the relapse, therapeutic resistance of AML, and then yet other groups, Ross Levine and others, have shown this for the progression of myeloproliferative neoplasms. So really more a general principle now in the field than the exception. And that, of course, shifts research questions. So we're now really asking what causes the stem cell subclonal diversity and what determines their dynamics, and what are the factors, and what can we learn from this also ultimately for our therapeutic approaches. And, you know, for that, I want to introduce the concept also of epigenetic heterogeneity. What I've showed you so far relied on genetic markers. And what I want to say, while easily tractable and detectable, there's more than just genetics to tumors. And that's not a new idea. A lot of labs have worked on this in the last 5 years. And we were particularly interested in this and in the context of transcription, in the context of stem cells and leukemia pathogenesis. And that's a more recent story I want to talk to you about today. And this is just to remind you what you've heard many times from Rob Singer, Rob Coleman, and others here at Einstein, that transcription is not a continuous process. And you know, there's a variety of molecular reasons for that. But what you can see, and I'm not going to go into details here, but what you can see here, if you look at the gene activity transcription at a single cell level and single locus level here, and this is in collaboration with Rob Coleman, you see that genes, they're on and off, they fire and they are silent. They fire, then they are silent, and that is how, you know, gene expression works. And if you look at this in an area under the curve graph, you can really see this. There's no such thing as a constant level of transcriptional activity, but it's really these on-off type phenomena that are characteristic for this process. So now that poses a lot of interesting questions that an MD PhD student in the lab who just recently went to start his residency at Harvard Brigham and Women's, Justin Wheat, wanted to address. And he really asked, you know, is the observed heterogeneity that we pick up in many of our and current single-cell RNA-seq studies, is this really true subpopulation diversity, or are some of these effects really driven by biophysical stochasticity? In order to address this, we collaborated with Rob Singer's lab and really adopted a method that Rob's lab had developed more than 20 years ago, single molecule RNA fish, and adopted it for the use on primary flow sorted hematopoietic stem and progenitor cells. And that's what Justin did. It took him about 2 years to get this done. And what this then allowed us was to visualize single molecules of RNA in our target cells of interest, including primary cells. And so this is how this then looks like. When you look at it, it's beautiful pictures. Each dot is really an individual RNA molecule, a single RNA molecule. And you not only get numbers, but you get subcellular localization, whether these molecules are in the cytoplasm or in the nucleus, and you get dynamics informations. You see at some points where these signals are clustered, then you know there's an actual transcription site, you know, where the gene is actually firing and where these RNAs are produced. And so we use this to look at a transcriptional network, the GATA1, GATA2, PU1 network, and we made some really interesting findings. Don't want to go into details here, but just cutting a long story short, we showed that all these genes or these transcripts were actually co-expressed in the same single cells, something that had been disputed by very recent, actually a bunch of very recent single cell RNA-seq studies. And just again, demonstrating you really need high sensitivity methods to come to these types of conclusions. But what we really wanted to understand, what is the dynamics of that process? And in particular, is there in stem cells, is there something like transcriptional priming, another very popular concept that had been introduced recently based on single cell RNA-seq studies? And to address this and study stem cells in a longitudinal manner, we adopted a method that was initially developed by Michael Elowitz's lab at Caltech. And you know how this works is you take a single cell, in our case, a single hematopoietic stem cell, and then you see how that cell divides and starts to differentiate. And then you look at the endpoint state and determine the transcriptional networks. And the idea is that if there is early lock-in, priming of the stem cells into one or the other transcriptional path, what you would expect is that closely related brother, sister, first-degree cousin cells would be more likely to be in a related transcriptional state, here depicted by blue, for instance, or red, but it wouldn't be very mixed. However, if that's not the case, if there's an early commitment, early priming, you would more expect a picture like this, where cells of both states are, you know, very mixed and matched, even if they're closely related. And so that's what Justin did. And you can see here a movie playing of one single stem cell that then divides and gives rise to progeny up to 46 over the course of 3 to 4 days. And then at the end of this, we performed the single molecule RNA FISH and determined the transcription states. And I'm cutting a long story short. What we found was the first option that I showed you, and that was that the transcription factor high states were paired with the uncommitted states really at all generational distances. So we saw the mixed picture, and what we concluded from that is that transitions to a high expressing state do not occur early and irreversibly in stem cells. So how do they occur? And in order to find that out, Justin repeated this experiment several hundred times and looked at the pedigrees, and every time determined the end product of this process. And then he matched the experimental data with models of transitions, and inferred rates of transitions between the different states. And that was a very close collaboration, again, here within Einstein with Aviv Bergman in systems biology. And what turned out very quickly in these studies was that irreversible models, unidirectional models, either irreversible chain or a source sink model that we tested, matched the experimental data very, very poorly. And what rose to the top were fully reversible models, and in particular, a so-called Markov chain between the transcriptional states that these stem cells shuttle in between. And there's some complicated mathematics behind this that then allowed us to really propose a quantitative hematopoietic stem cell transcription state transition model. And what you can see is all these states in the stem cells are fully connected and reversible. And in fact, while the highest likelihood is that the cells maintain their identity from one to the next division, there is some probability that they switch identities, but very importantly, even if they reach one of the endpoints or the later stages, like the PU1 high state on this edge or the GATA1 high state on this edge, there's a very significant probability that they actually go back to some of these intermediate states. So again, there is a very plastic and probabilistic component to this. And to summarize this, we actually think that this is in fact a feature of stem cell biology, in the sense that this noise and this fluctuation actually serves an important purpose, and that really deterministic cellular output can arise from gene expression patterns that are probabilistic and noisy in nature, because automatically, if you pull a cell out of this, the stem cell will essentially replenish the entire plasticity and this chain again, and thereby maintain the functionality of the stem cell pool. And so we propose that this intrinsic stochasticity of gene expression really facilitates rather than impedes the concomitant maintenance of transcriptional plasticity and stem cell robustness. So now, this is really the main takeaway, is that we think that transcriptional plasticity and heterogeneity is really the natural space that stem cells reside in. So all the heterogeneity that we see and pick up, there's nothing unusual about it. This is just how these cells are, in particular stem cells, where many transcriptional programs are not robustly established and at very high levels established yet. Now, what does it mean for our understanding of malignancy? And I don't want to, talk about this too much in the interest of time, just refer you to 2 review articles that we wrote on this topic. Just in a nutshell, we think in addition to the genetic progression and genetic mutants and subclones that get selected for, there is an orthogonal kind of system driven by transcriptional state plasticity, and that adds further complexity to subclones that emerge during disease pathogenesis and treatment. And then maybe on the right side, a very important point. Now, if you think about subclones, here depicted by these hills and their relative fitness, in the z-axis here, all of this very much depends also on extrinsic selection pressures. And that is something that Dr. Verma will talk about it quite a bit in his talk. So mutants and subclones, they're not good or bad or aggressive or not so aggressive, just intrinsically. It all very much depends on what are they exposed to in terms of inflammation, environmental exposures, drug treatment. All this, shifts the subclonal competition of these subclones. Now, again, coming back to the scheme that I now essentially told you is wrong, what that means is really that these pre-leukemic stem cells are incredibly heterogeneous. And we're not just dealing with one clone that we have to target, but with an entirety of like entire bunch of subclones. And you can say, well, this is completely hopeless. How are we going to tackle this daunting heterogeneity? There is approaches though, and that's what I want to show you in the remaining a few minutes of the talk. And we have done very significant and systematic efforts, both in mouse systems as well as in sorted pre-leukemic stem cells from humans, and really identified a lot of targets in the last few years. And you'll hear about this from Dr. Verma as well. There's 2 I want to briefly talk about: MDMX and IL-1RAP. So the first one is an immunotherapy target that we discovered almost 10 years ago. Was another PhD graduate student in the lab who discovered this, and then another PhD student who added a mechanistic angle and new mechanistic insights to this, how IL-1RAP works in hematopoietic stem cells. And obviously, you can imagine if you have an immunotherapy target, that target doesn't care about subclones. If it's expressed on all these subclones, you will be able to target them and take them out in their entirety. So the second I want to talk about in the last few minutes are transcriptional regulators. And here I want to focus on MDM4, also known as MDMX. And that is one of the endogenous inhibitors of the p53 gene. And what you can imagine is that if MDM4, MDMX is upregulated, it will inhibit p53 activity, thereby reduce apoptosis induction. And you can imagine that that would then lead to the generation and survival of more and more aberrant subclones. And so we were initially surprised, but then thinking about this, not so surprised, that we discovered a few years ago that MDMX is particularly high in stem cells from patients with AML and also MDS. And if you look at this data from TCGA here, this is AML. These are all the other tumors or many other tumors in the database. You really see MDMX sky high in AML in AML only. We were able to show that it can be a therapeutic target in preclinical xenograft models, and this also led to a variety of clinical trials, several of which we were part of here at Montefiore. And what we then really wanted to ask, and this is a very recent paper, is what is the mechanism of this? And for that, we collaborated with Gigi Lozano at MD Anderson and looked at MDMX transgenic mice. And what turned out very quickly is that MDMX overexpression induces serial replating capacity in cells, but also leads to a competitive stem cell advantage when you compare it to co-transplanted wild-type stem cells. And then in addition, we were wondering now, can MDMX overexpression really push pre-leukemia over the edge? And that's a question that a very talented postdoc, Koki Ueda, asked at that time. He now just started his own lab in Japan about a year ago. And he generated 5 new mouse models of pre-leukemic aberrations, a variety of pre-leukemic aberrations that don't lead to acute leukemia by themselves, and asked what happens if we cross the MDMX transgene into these models. And without going into details, he found that in every single model that we looked at, the elevation of MDMX really pushed it over the top and then led to acute myeloid leukemia formation. And then the cool thing was, and of course we were wondering, is this now just a funny mouse thing that's happening here, or is this relevant in patients? And so we again teamed up with Amit Verma and also collaborators in the UK, Jackie Boltwood's group. And what you see here is these are patients with MDS, and if you dichotomize them into MDMX high and low expressors, you see that the MDMX high expressors are very likely to progress, transform to AML within a very short period of time, within a few months normally, while the MDMX low expressors do not. And this is really specific to the progression to AML, not other deaths or other causes of death where essentially you see no difference. So really likely, nicely backing up this, this leukemia or pre-leukemic to leukemia transition concept. And that's the conclusion here. I'm wrapping up. So MDMX increases competitiveness of HSC, acts as a pre-leukemic to acute leukemia switch mechanism, and clinical correlative data suggests relevance of this pathway in patients. And now, of course, the really big question is now, can we use MDMX inhibition ultimately as a prevention strategy, precision prevention strategy for patients with MDS or at a pre-leukemic stem cell stage? So, I hope I've convinced you that pre-leukemic stem cells are very important, that it's not entirely hopeless despite their heterogeneity, that there is some targets that are emerging and that are targetable and clinically actionable. And I just want to leave you with some final thoughts, and that is really, the pathogenesis of myeloid malignancies is a fundamentally polyclonal process. Bulk cell genomics seems insufficient to predict the precise course of disease development, progression, and lasting therapeutic response. Non-genetic mechanisms contribute to the heterogeneity. We have now candidates of these pre-leukemic to leukemia trigger mechanisms. And then also cell extrinsic selection mechanisms play a key role. And you'll hear a lot more about this from Dr. Verma in his talk. And with that, I just really want to close and just say or reiterate again what was said in the beginning and in the very nice introductions by Dr. Ozoa and Dr. Tomaselli. All of this only possible within this terrific and highly collaborative Einstein environment. Phenomenal lab, trainees, students, postdocs, technicians in the lab who have made all this possible. Strong support from the Cancer Center. And then, of course, Amit Verma and the Division of Hemato-Oncology, and many others there. And I'm just having a few names here. Amit will mention the entire team, Aditi Shastri, Yanis Manzares, Mendel Goldfinger, who were very involved specifically in our studies. And then many collaborators within cell biology, Rob Coleman, Rob Singer, Art Skolci, Britta Will, Kira Gritzman, Winfried Edelman, and others, Aviv Bergman in systems biology, highly specialized core facilities, and then our outside collaborators. This is my lab. With that, I'll stop, and I thank you very much again for your attention. Thank you all so much.
Perhaps we have time for a quick question or two if anybody has one. Maybe after the second. So Uli, thank you very much. That was terrific. It's now a pleasure for me to introduce the second of the dynamic duo, and that's Dr. Amit Verma. He is a professor of medicine and of developmental and molecular biology, associate director of translational science at the Cancer Center, and a director of the Division of Hemato-Oncology at Montefiore and Einstein. Amit earned his medical degree from the All India Institute of Medical Sciences and did postdoctoral training in internal medicine at the University of Illinois, Chicago, and a fellowship in hematology oncology at the Robert Lurie Comprehensive Cancer Center of Northwestern. After 2 years at UT Southwestern, we were able to recruit Amit to Einstein as an assistant professor in oncology and developmental molecular biology, where he quickly rose through the ranks to full professor. He's earned national and international recognition and a number of awards, and too was elected to the ASCI in 2015. His lab's been involved in studying the pathogenesis of myeloid malignancies, such as MDS and AML, and research from his lab has elucidated the critical role of various signaling pathways in MDS, and the work has directly led to therapeutic targeting of these pathways in clinical trials of MDS and AML. In close collaboration with Dr. Steidl, Dr. Verma has defined stem cell alterations in MDS and AML that have translated those findings to the clinic. He's worked for a number of years as well on the World Trade Center exposure of first responders and the detection of clonal hematopoiesis and leukemia in these patients. And more recently, as many have done, he's conducted clinical studies in hematologic malignancies and examined the effects of what we've all been living through for the last two and a half years, COVID-19, as well as environmental exposures on outcomes and pathogenesis of cancers. So I welcome Amit to the podium. His talk today is entitled Therapeutic Targeting of MDS and AML. Amit.
Thank you very much, Gordon, for the very kind introduction. And thank you to Dr. Ozwa for this tremendous honor and the selection committee. I wanted to focus in this brief talk on the collaborative nature of all that we have accomplished here at Einstein and Montefiore, and some translational aspects of the work that Uli talked about, and some more upcoming things that we are looking forward to. So a little introduction about the disease that I work on. This is a disease called myelodysplastic syndromes, or MDS for short. It's actually a collection of various cancers, neoplasms, malignant hematopoietic disorders. And the common theme is low blood counts. So there's bone marrow failure, ineffective hematopoiesis, and the patients get low blood counts. And that causes most of their mortality and morbidity. Now, there is a tendency to progress to acute myeloid leukemia. A third of the patients with MDS become frank leukemias. And the overall incidence of this disease in the US, the real number is about 50,000. And this number actually keeps going up as your population ages and as we have better ways of detecting early signs of disease. Now, this disease is predominantly a disease of the elderly. You can see the sharp spike in incidence of MDS as our population ages. And also, this is a disease that actually is a pretty serious disease. This slide shows you that the survival of patients with MDS is not very good. So this is a stage-by-stage comparison with lung cancer, which is universally recognized as a serious disease. And you can see we divide MDS into lower risk forms and higher risk forms. And these are overall median survivals. So even with lower risk MDS, we're looking at a pretty bad survival. These low-risk forms of MDS don't necessarily transform to frank leukemia. But what causes mortality in these forms is low blood counts. So you have neutropenia, propensity to die from infections, you have thrombocytopenia, propensity to die from bleeds, and anemia, which leads to heart failure, iron overload. So when I started in Einstein, we had a simplistic model. Uli talked about aberrant clones of hematopoietic stem cells. And the question is, what makes these aberrant clones grow over time as we age? And we focused on inflammation. So there's a marrow inflammation that a lot of people now have postulated as the cause of the growth of aberrant stem cells in MDS and AML. The inflammation also leads to suppressing normal hematopoietic stem cell clones. And the abnormal clones, they're able to resist inflammatory cues. They're able to resist apoptosis and grow preferentially. Give them time, they eventually can transform to MDS phenotypes. And when they acquire second, third, fourth hits, can also transform to acute myeloid leukemia. So can we intercede at these disease stages, inhibit inflammation, and cause benefit to the patients? And I'll show you some examples. So when I first started, we focused on a broad MAP kinase that is triggered by a lot of these inflammation associated cytokines, p38 stress activated MAP kinase. We showed that it's quite heavily activated in MDS bone marrows. And this work led to a couple of clinical trials. One was with an early p38 inhibitor that showed some signal, but wasn't really taken forward in further trials for a variety of reasons. And more recently, with a drug, ARI-614, which was tested in MDS patients and actually showed some very interesting responses in refractory cases. And we are trying to work out the molecular mechanisms now. But more currently and more importantly, we have focused on the role of a very important cytokine, TGF-beta, transforming growth factor beta in the pathophysiology of MDS. Now, this growth factor is being studied a lot by many talented investigators, and it's a very large family of ligands. They have some common themes. There's a set of receptors, which we call as activin-like kinase receptors, or ALKs, which then phosphorylate intracellular proteins called SMAD proteins. And these SMAD proteins can associate with each other and go inside the nucleus and act as transcription factors. And they cause very important effects on hematopoietic stem cell proliferation, as well as differentiation. So many years ago, we showed for the first time that the SMAD2 and 3 transcription factors are very heavily activated in MDS bone marrows. These are patient bone marrows. You can see the brown stuff in the nucleus. This shows heavy intensity of SMAD activation in MDS compared to anemic controls. And this led to a lot of translational efforts. We also studied a mouse model that constitutively produces TGF-beta and mimics some signs of MDS, leads to anemia and thrombocytopenia in these mice. And we've used this mouse for a lot of translational studies. The first drug that we tested and showed had potential in MDS was an oral TGF-beta inhibitor called galinosertib, made by Eli Lilly, which actually went into clinical trials. This was a clinical trial of this drug, which was conducted primarily in Europe. And for heavily transfused patients with MDS that were requiring more than 4 units of red blood cell transfusions every 2 months, it actually showed a signal. 37% of them benefited from this drug in terms of hematological improvement. HI means their blood counts rose beyond a certain level. We did a lot of correlative science with these samples, and we showed that when you look at the responders to this drug, these had a large number of circulating hematopoietic stem cells that were arrested at the hematopoietic stem cell stage, that were unable to differentiate. So they had a differentiation block. I have to say, the ability to do all of these correlative sciences was completely in part due to the infrastructure that Uli built, on the sixth floor of Channon Building with the support of Einstein and Montefiore resources. And we have used this stem cell lab for many such collaborative projects, which are still ongoing. Okay, so a lot of studies we did, collaborative studies, and this diagram just summarizes a lot of them. There are these microRNA circuits that can actually inhibit an endogenous inhibitor of a lot of these TGF-beta pathways, which is called SMAD7. And when this goes down, the pathways, the TGF-beta ligand activated pathways constitutively start firing up. And we have showed in multiple studies that when you have hyperactivation of SMAD2 and 3 pathways, you end up with anemia. You end up with ineffective hematopoiesis and low blood counts. So translationally, how can we reverse this process? I've shown you data with a drug that blocks the classic TGF-beta receptor. Now, a few years ago, we have worked with a small biotech, Acceleron, not so small anymore, that developed another TGF-beta ligand trap. This is a drug called luspatercept. It contains the extracellular domain of an activin receptor 2B molecule fused with an IgG Fc domain. So this drug was developed for osteoporosis. When they gave it to human volunteers, they found that their hemoglobin went up by 3 points. Just one administration of the drug. Same time, our paper with SMAD2 came out. This drug actually is a very strong inhibitor of SMAD2 phosphorylation. And we did a lot of studies. This is the net result of a lot of preclinical work. It turns out that in MDS, there is ineffective erythropoiesis. There is actually a deficiency of late-stage erythroid progenitors. And when you treat preclinical mouse models or in vitro samples with this drug luspatercept, it increases late stages erythropoiesis in these models. Now, this drug actually went to phase 2 trials, which were positive, went to a phase 3, which we also participated in Montefiore, the Medalist trial. And the success of this trial led to an FDA approval in 2020. And these were the results. So this was a randomized trial, luspatercept versus placebo in patients with MDS. Luspatercept led to a 38% response with transfusion independence as compared to 13% in the placebo arm. And this shows you the hemoglobin tracings. This is luspatercept, and this is placebo. So this was the first drug, actually. It's a pretty hopeless disease, these diseases that we work on. There are not too many curative strategies, even at the present time. But this drug did lead to improvement in hemoglobin and led to an FDA approval. Now, Uli's talked about the importance of stem cells. And this case, this is a local Bronx case of a car mechanic who presented to our clinic in Monty with bad anemia. You can see his hemoglobin goes down. We give him a transfusion, goes down again. We put him on a clinical trial with a drug called azacitidine, which is an FDA-approved agent. And his hemoglobin improved tremendously. You can see he became transfusion independent. But at the same time, we were doing bone marrows on him. And his leukemic stem cells and pre-leukemic stem cells actually did not go down. These two tracings show that the aberrant stem cells actually were hanging around even when he was in a remission, and they actually went up. And 2 months later, he has a bad relapse. Unfortunately, doesn't make it. So the important point that Uli emphasized was, unless we target leukemic stem cells, the disease-causing stem cells, we will never have long-lasting remissions in this disease. And we've developed a few things translationally. Dr. Shastri, who is now an outstanding independent PI, is working on inhibiting the transcription factor STAT3, which is overexpressed in leukemic stem cells. codes for a bad prognosis. She's working for a variety of very innovative approaches to inhibit STAT3 in preclinical and clinical studies. We worked on the role of this innate inflammation molecule, interleukin-8. We showed that it's high in MDS. This receptor codes for bad disease, and patients with MDS that have high CXCR2 have more transfusion dependence. And this has led to a translational effort, which I'll talk about. And in the same theme, Uli talked about IL-1RAP. IL-1RAP can actually signal through a very conserved pathway, the MYD88 adapter, which then phosphorylates the IRAK-TRAF6 complex. And we showed that patients with MDS actually have a longer isoform of IRAK4. And this can be targeted with a small molecule, CA4948, which has now gone in the clinic. And the initial data looks very intriguing with some very nice responses. So the emerging theme is inflammation perpetuates these diseases, and specifically innate immune signaling, IL-1RAP, IRAK, IL-8, all of these pathways can increase the MDS and AML phenotypes and support the growth of stem cells. And there are a variety of approaches, including some in biotech that are targeting some of these molecules with monoclonal antibodies. Lastly, I'm going to briefly tell you about another unique environmental exposure that I've been fortunate to work with. And this collaboration is again, a testament to the close collaboration between Montefiore and Einstein. So after 9/11, the buildings came down. There was a massive exposure to the first responders and other local residents to this World Trade Center dust cloud. And Dr. Dave Prezant is a pulmonologist at Montefiore. And he has a whole team of very talented biostatisticians, including Dr. Z. Govans, who have been analyzing outcomes in first responder firefighters and EMS workers who were exposed to the 9/11 disaster. They published a seminal paper in Lancet in 2011 that showed that there was an increasing incidence of some malignancies in these first responders. We started working together and also enlisted a collaborator in Sloan Kettering. We were funded by the V Foundation to set up a bank of samples from these firefighters. So over the last few years, we've collected blood from a lot of FDNY firefighters and also, now 300 controls that are firefighters who have not been exposed to the 9/11 exposure. So this is the bank, fractionated blood, a lot of cool things we can do with it now. The first study we published was a study looking at early signs of multiple myeloma, which is another blood cancer. And we found that the odds of getting myeloma precursor in the first responders was about 2 times more than reference population. And it was not that they were getting it more, but they were actually getting this myeloma precursor at a younger age. So the median age in the first responders was 10 years earlier than what you see in the general population. Also, we found cases of full-blown myeloma in the 9/11 cohort, and there was something very interesting. Biologically, 55% had light chain disease. Usually we see less than 10%, and we're trying to dissect out what's really happening here. More recently, we've tried to work the mechanisms, how this actually happens. The World Trade Center dust was incredibly genotoxic. A lot of chemists and labs have tried to look at the constituents of this dust, and you find a lot of really toxic chemicals, a lot of organic polyamines, dioxins, asbestos. And the first responders, they didn't have masks. They found that the masks were actually very huge and laborious while they were doing the rescue effort, so they took them off. So they were inhaling this toxic dust. we recently published results of sequencing 481 first responders compared to 250 controls. And these are mutations that are seen commonly in MDS and AML and clonal hematopoiesis. You can see the difference between the first responders and the control cohort. And we're trying to now do broader studies and figure out, how many of these mutant cases actually have signs of or phenotypes of actual frank diseases. I'll quickly go through in the interest of time. We have dosed mice with this World Trade Center dust and replicated some of these findings at the stem cell level as well as at the genomic level. So what's the future plans? We're now sequencing the whole cohort. We're going to correlate this with exposure to tease out what really is the genetic predisposition. Why do some people get this and why don't other people get this? And try and make up a clinic, here at Montefiore, which will try and look at these cases that are otherwise healthy, but have these big mutant clones that we can detect in their blood. What can we do for primary prevention? Actually, these diseases have a strong signal for cardiovascular diseases, for inflammation, for diabetes, for lung diseases. So maybe we can do primary prevention for other organ systems, not necessarily, leukemia. So a lot of people to thank. This is a tremendous team effort. Very collaborative science. Firstly, Dr. Ozwa and Dr. Tomaselli for their continued support all these 16 years. The huge infrastructural support, the core services, we can't do anything without this. Dr. Chu, who's, in the short span of time, taken the cancer center to, really a dynamic height, and we're using a lot of the things that he's brought to Einstein, and Dr. Goldman before him. And, the people who've done all of this work, I'm just a representative presenting the work that all of the present lab members and the previous lab members have done. Uli, I was lucky. He came a few months after I joined Einstein, and it's been a wonderful partnership. John, who's built a tremendous epigenomics facility. Britta, outstanding PI in her own right. We've continued to collaborate well. I mentioned Aditi's, efforts with STAT3. Teresa with her modeling, Everest, chemical biology. There's so many people, and this is just a few of them. And last but not the least, the tremendous faculty we have in the clinical domain in Montefiore. Kira, Alex, Mendel, Nishi, Swati, Noah, Aditi, and Yanis, who have, they're not just outstanding clinicians, but also very, very academically inclined scientists, clinical scientists. We have, in the last few years, built a tremendous clinical infrastructure. We're doing a lot of cell therapies, and these chimeric antigen receptor T cell therapies in the Bronx. And the guiding force is to bring these therapies to our local patients in the Bronx. A lot of firsts, efficacy in rare diseases, in underserved populations. These guys published the first report of efficacy of CAR-Ts in minorities. We are dealing with a patient population that's 85% minorities. And now, with Dr. Ozwa's support, he facilitated a nice philanthropic gift. We're trying now to build in-house CAR capabilities that hopefully we can bring to fruition in the next year or so. So thank you again, and happy to take any questions.
Perhaps questions for either Amit or Uli? There aren't?
Hi, Amit. I have a great question for you. In the study in the bone patients where they were given the TGF-beta trap antibody and their hemoglobins went up, does that imply that even without obvious inflammation, there's a baseline effect of TGF-beta? And then the question is, how about all the other cytokines? And, can the system upregulate by upregulating TGF-beta receptors instead of just the ligand? How do you think about it?
That's a great question. The trial was done in healthy human volunteers, and their hemoglobins, which was normal, went up to polycythemic ranges. And that tells you, the whole process of hematopoiesis is so tightly regulated. And we don't really know these important clinical sort of regulatory mechanisms. But I can tell you that this particular drug does increase tremendously late stages of erythropoiesis, even in normal healthy people. And the mechanism is still being worked out. Part of it is binding to a molecule called GDF11, but that's only part of the story. And we're trying to find out what are the off-target effects that the drug may be doing.
Very nice to both of you. Question about TGF-beta. Do you know if all ligands operate the same in this role, or there is some distinction in the ligands?
It's a great question, Julio. So it's a lot of unpublished work, which we're doing to address specifically this question. It seems like the classic TGF-beta molecules tend to act more early, and the GDF activin B molecules might be regulating later stages more. And that is partly, even though SMAD2 is the dominant transcription factor, but its association partners and where it binds on the genome may be different with different ligands. And that leads to translational application of possibly combination therapies and so on and so forth, which should happen sometime in the future.
Richard.
My question is for you. And so given the elevated expression of IL-1RAP and MDM8 in the multiple clones in these stem cell population, where are you guys with, or other people with developing inhibitors and their testing for those particular?
Yeah, it's a great question, Richard. So let me start with MDMX. And we actually helped a preclinical drug development study there. There is a small, biotech company in Boston, Cambridge, that has brought the first dual MDMX, MDM2 inhibitor in the clinic. And so that is a way to target this pathway. I didn't go into the mechanistic details. It may be required to combine that drug that only targets the MDMX p53 interaction in addition with a beta-catenin inhibitor, because that's an unexpected effect that we found, or mechanism. So it's not exclusively p53 mediated, but there is ways to target this. And again, the ultimate goal would be to upregulate, reactivate p53, and then which would lead to an elimination of aberrant subclones. The second question is IL-1RAP. So that was really a very new target that was discovered again about 10 years ago. There is one company, again, a small biotech in Sweden, that has developed an IL-1RAP directed antibody that is now in clinical testing. Although, if I understand the press releases and everything correctly, they are mainly going into solid tumors at present, where this has also popped up as a target. And then there's multiple other companies who have assets at various preclinical stages, also directed against IL-1RAP. So I think we'll see a lot of clinical action in that space, and hopefully including in MDS and AML, and maybe even before at this clonal hematopoiesis stage very soon. So as you know, it takes a long time to go from target discovery to getting something into the clinic. But I think, two companies are already there, and I think there's much more coming for both targets, actually. So we're pretty excited about it and see what happens. And, the second thing I just want to say is, I think it'll also be important. So the clinical community has changed in the last 10 years. And I think everybody now recognizes we have to intervene early. This idea of going in before the bad things happen. This is now 10 years ago, nobody would have listened. And I did propose it 10 years ago and nobody listened to me. And at these more translational meetings. Now that has completely changed because the field has moved. And I think this will also make a big difference because a lot of drugs who quote unquote fail in very late stages, they may actually work if we try it early enough and, you know, at the stem cell level before everything has gotten completely out of control already. And I think that's another very important trend I think that we're seeing and that hope will continue, you know, in the translational space.
What do you mention?
Yes.
Look, Oli, I have a question for you.
Yeah.
I'm trying to understand this polyclonal. Is this a driver mutation and then the cells go back and forth on the Markov, and so they all show up as polyclonal that way? Or are there multiple driver mutations, in which case is there then some underlying genomic instability that's giving rise to them? Because I'm sort of the Mo Knudsen second hit.
Yes. Yeah, no, I think it's a fantastic question. And I think the entire field has trouble, wrapping our heads around it, if you will. What has been found is that even if you look at normal hematopoiesis and as we age, a lot of these clones do exist. So there's a lot of mutant stem cells in like just all of us as we're sitting here. And, so the real question in the field is, why do they not come out? In fact, in the majority of people, they're well detectable. If you look with sensitive enough methods, why don't they come out? Why don't they progress? Why don't they lead to malignancy? And I think it's really a great question. My preliminary conclusion is, and that's not totally accepted in the mainstream yet. But I would say it's, the genetics is overrated. The genetics as a standalone is overrated because clearly the genetics events alone do not lead to transformation in everybody and in each of us. So the question is, what is it? And, genomic instability, of course, is one possibility, that there's just then more and more genetics happening and that brings it over the top. But, I think it's more than that. And I think it's the epigenetics. And some of that, has to do with this shuttling that I described, including at the transcriptional level. There's also very good data with other epigenetic mechanisms, including methylation, that plays a role on top of the underlying genetics. And then there's mechanisms like Amit described that you say, well, maybe a clone, just the genetics, it doesn't matter, right? But then you expose that clone to a certain challenge, an environmental stress, inflammation, any type of extrinsic pressure that then gives that clone that under normal circumstances and in young people would not come out, then gives it an advantage to really achieve clonal dominance and then turn into a malignancy. But, in all honesty, all this is just pretty hand wavy right now. I think the jury on that, all of that is still out there and it's current, a subject of intense research, but I think that's where the field is focused on right now. But I think it's the big question in the field, exactly what you just asked. Hilda.
I have a question for so the two, reinforce the concept that inflammation, stem cell, homeostasis, oncogenesis are fine. So I want to ask a question. You show very nice genetics in Wall Street settings that way. Do you have plans for what those might be already started to analyze the immunological signatures in these settings? This is a very sizable cohort that could tell us either it's correlative or maybe the origin of the mutation. You look at the bottom, which means white blood cells, B cells and T cells are contributing, but are they talking about memory patch?
Yes. No, that's a great point, Hilda. We haven't done it, but we have the cells. These are all peripheral blood samples, so we have a bunch of mononuclear cells. If somebody wants to collaborate, we're open. But it's a great point. The immune system contribution in suppressing certain clones and keeping them suppressed has been relatively understudied, partly because we don't know these new antigen landscapes and things like that, but still, it has to be important.
Another question? Yes.
All right, I'll do it. So pursuing the question of genetic versus more than genetic. Your work showed nice variation among stem cells as they grew in the dish. Amit raised the question of inflammation. In the middle between the two is, do you see the same behavior in expanding stem cell population in its correct environmental niche, or is it much more stabilized and responsive to the local environmental there? And the consequence of that is that could this disease not be a cell autonomous genetic disease, but rather a mismatch between essentially normal stem cells and an aberrant environment?
Yeah, absolutely, Moshe. It's a great question. And I mean, let me start with the first one. So, inflammatory signals and that, is an absolute, right on point, the question. So what we have done in that respect is, I mean, first of all, what I've shown you, it's kind of so, complicated technically. So we have exposed these stem cells under the same conditions, still in vitro, but exposed them to inflammatory cytokines. That's stuff we're doing right now. It's still ongoing, but I can tell you that, we do see changes in, the transcriptional dynamics and the state transitions. We do see changes, and that's very exciting to us. But nonetheless, what you're saying is absolutely right. We want to do this in the in vivo setting. Now, the problem with that is that there you need methods to do this live, in living cells, ideally even in a live animal. And that's, we have a very active collaboration with Rob Coleman's lab right now. We've now knocked in both RNA tags, but also protein tags that will then allow us to do single molecule imaging in live cells in a mouse, both at the RNA level, but also at the protein level, single molecule or single particle tracking in live animals. That's an active collaboration. It's ongoing. That's exactly where we are going with this on the science side. Now, your second question, like more general, I 100% agree with you. And that's, I went over this very quickly. I referred to this one review that was written by another phenomenal MD PhD student in the lab, Emily Schwenger, who discusses this, what does it mean? You have aberrant phenotypic clones, and you could genetically define them, epigenetically define them, any combination thereof, right? The relative fitness of those clones is not determined by just cell intrinsic, genetics, epigenetics, whatever it is, but is determined by, in what niche, in what environment do they operate in, and what other subclones are around at the same time, including like a wild type, like unmutated, completely normal stem cells. And I think the entire field is shifting to these types of questions, not looking at the cells as a standalone anymore, but in the context of, of the niche, of different challenges, in the context of aging. There's a lot of things that happen during aging, including in the niche. And a lot of our, newer faculty, I'm just looking around, including ones we have hired fairly recently, are focused on exactly these types of questions. I think this is exactly where the field is going and where the field has to go, I think. Absolutely, the right question.
Perhaps we have time for one or two more questions. Jackie?
Yeah, so my question, fantastic talks, both of them, and I'm not a hematologist, I'm a specialist. My question is related to all of these factors that influence that clonality and also like positive versus negative shift of that heterogeneity. Where do you see vaccines from, right? Because we learned during COVID so much about the epigenetic changes that live vaccines, particularly BCG, do, but we also know that measles vaccine and other infections actually, and all of this dynamic could start very early. Is it good or not? And that might be an opportunity to do pre and post vaccination with live vaccine. Where is your thought of the influence
yeah, maybe I can start and Amit can take over. So I think it's a good, an important thought. I think there's no evidence, at least to my knowledge, that any type of infection or even repeated vaccination or single like infections that we're all exposed to, right? I mean, all of us, in our families, and I mean, we have infections like all the time, and I don't think there's any real correlation. There is some correlations with more chronic inflammatory and conditions. They have a slightly but detectably higher risk of leukemia incidence, MDS incidence, progression or development of these diseases, but it's very mild. But I don't think there's evidence that like single, either vaccination or infectious stimuli really do something that's bad. I don't know, Amit, you can comment on this more, but.
There's a signal, I think, that will emerge. There've been 2 papers published with COVID now that show a slightly higher rate of clonal hematopoiesis in COVID patients who had severe disease. And there is a little cell autonomous element to the story. The commonest mutation in MDS are genes that affect RNA splicing, not P53 or KRAS, but things like SF3B1, U2A1. And it turns out that the IRAK4 protein, which is a transducer for inflammation, is aberrantly spliced, and you have more of the active form in these cells that have the mutation. So they respond more to inflammation, like interleukin-1 beta. So I think it's a very important point. We'll see more now as these concepts are emerging.
Leo, did you have one last question?
I have a burning question about this proposal and it connects with some of the other questions. Has anybody looked at these transition clones or the rate at which CH progresses in patients treated with rheumatoid arthritis?
Yeah, it's a great question. There are people doing it. There's every week there's a new paper linking clonal hematopoiesis with another inflammatory condition. I don't know if the direct association with rheumatoid arthritis has been published yet, but there's osteoporosis connection, diabetes, SLE. So, there is now the bigger question, Julio, the chicken and the egg. So all of us, when we age, have these mutations, right? There is a school of thought that says that some of these mutations actually make your monocytes and macrophages more secretory, like TET2. And then there's a school of thought that inflammation comes because you have rheumatoid arthritis, and that supports the growth of this clone. So what comes first? We don't know that.
Great. I think we should wrap this up. I want to thank Dr. Steiner, Dr. Penny. Dr. Stanley, the Faculty Interactions Committee, Dr. Oswald, and all of you for coming today. Thank you.